Hello, BIO178. Welcome to the overview, the background information video for this Week 8 lab, where I will give some information about the data set we're working with. And just for reference, this information I'm giving you has actually been modified from a talk I gave in January at a plant and animal genome meeting. So if you're curious, this is a little bit of a taste of what conference talks are like as well. So this talk I gave was called Transcriptome-Wide Gene Expression Differences in the Intertidal Mussel Midalis californianus. So that is the study organism that we are examining today with our data. That's pictured here in the bottom right. That's a big mussel bed. So I need to acknowledge that this data we're working with today is not just because of me. There are several co-authors I have on this study with me. One person who has helped me with this data set is Florian, who's pictured here. He's actually an alum from Sac State. He was an undergrad in my lab last year, and he is currently a PhD student at Penn State. And this work was a continuation of work I did during my postdoc with Dr. Wes Dowd and his other previous postdoc, Dr. Rochelle Tanner, was also instrumental in helping us get this data. So for a little bit of background about the intertidal zone itself, organisms in the intertidal zone experience what we call temporal environmental extremes, and that's because of the timing of the tides. So in all of these pictures here, these different organisms are submerged or underwater during high tide when it's relatively cool. And then during low tide, when the tide goes out, all of these organisms are now exposed to air, including the mussels shown down here on the bottom left. So when these organisms are exposed to air, if that low tide is occurring during a warm afternoon, for instance, when the sun is shining, these organisms have to deal with not only potentially desiccation or drying out stress because they're not underwater, they also get very warm. We also know that many organisms, especially those that are sessile or that attach to a surface and then don't move, like the mussels, they also experience what we call spatial variation in the intertidal zone. So even within a single intertidal site, just a few meters away, there can be very large differences in temperature. So this is a picture of one of the field sites I've worked at. This is at Hopkins Marine Station in Pacific Grove, California. It's right by the Monterey Bay Aquarium. If you've ever been there, like where this picture was taken, just a little bit off to the right is where the Monterey Bay Aquarium is. It was a great place to work for a summer. So at this field site, we have a low intertidal site where these mussels that live there are relatively close to the water, so they get what we call wave splash. That wave splash keeps them relatively cool. Versus at this higher intertidal site that's farther away from the waterline, higher up the shore, those mussels aren't covered by water as often because of the tidal cycle, and so mussels at those sites are hotter. And we've actually quantified that, or I haven't quantified it, but previous people in the Dowd lab have. So this is a temperature record. We have the mean temperature on the y-axis, summer days on the x-axis, that high intertidal site's in red, that low intertidal site is in blue. And we can see that there are several days where there are these really huge differences where that high intertidal site reaches, here we're getting up to over 30 degrees C, whereas that low intertidal site doesn't even hit 20. So there are these huge, at least 10 degrees Celsius differences in temperature exposure between those two sites. That's a really big difference. There's another one for us. And we know that those temperature differences have effects on these mussels at these different sites. This graph, what we're looking at is DNA damage in the gill tissue, so you can specifically dissect out the gills of these mussels and quantify the amount of DNA damage that's happening in these mussels as a result of these environmental stresses. So we've talked previously about how under heat stress, one of the things that happens is proteins become damaged and denatured. They don't fold properly. Another common result of heat stress is damage to the DNA itself. And so in this graph, what I did is quantify the amount of DNA damage in muscles at both the low and the high intertidal site. Don't worry about the different colors here in this graph, but overall what we're looking at is that overall the amount of DNA damage at this high intertidal site is significantly more than the amount of DNA damage from muscles at this low intertidal site. And why do we care about this? So a couple summers ago, you may have seen in the news, there were these very dramatic headlines about mussels being, quote, cooked to death in their shells. And this was in the summer of 2019. We had really warm weather, and especially up in Bodega Bay area, there were tens of thousands of mussels actually died on the shore because they got too warm. They experienced really severe heat stress and they died. And we care about this because mussels are considered, quote unquote, a canary in the coal mine for climate change. And mussels are really important for that intertidal ecosystem because they provide habitat, they provide food for a bunch of other organisms. So if we're seeing these mass mortality events in these mussels, that's going to have far-reaching effects on other organisms, other species in the intertidal habitat as well. So we want to understand better the mechanisms that are causing these physiological differences, this DNA damage, and also this mortality. So that's the main thing the main question here related to the data set we're working with is what are the gene expression differences underlying these physiological differences, like physiological meaning DNA damage, for instance. So for the experimental design, the data that we're working with today has been produced from this experimental design, which is a little bit complicated, so bear with me. It took me a while to actually understand what we were doing. So one group of mussels was taken directly from the field. We're calling them the field acclimated group. One group we actually brought back into the lab and kept them in common garden conditions for 1 month in an aquarium system. We sampled some mussels from that common garden. Another group we put back out into the intertidal at this relatively low stress outplant low site. And another group we put out back into the intertidal at this relatively high stress, the warmer outplant high site. And just in terms of sample sizes and abbreviations, we had roughly 10 mussels from each of those different treatment groups. And the data set we'll be working with, you're actually going to see these abbreviations here in the middle column. The field group is abbreviated FA, common garden CG, outplant low OL, and outplant high OH. So once we had the actual samples, we got the samples from these different field and lab treatments. I extracted RNA from the gill tissue. This is what a mussel looks like when you open it. So normally the mussel keeps itself closed. You can actually slice one of their adductor muscles to open the mussel. And this is a mussel that already has its gill tissue removed. Normally that gill would be right here. I took a picture because this is actually a pearl that I found in a mussel. I've dissected lots of mussels. This is the only one I've ever found. So once I extracted RNA from the gill tissue, we did RNA sequencing, and at that point I basically left that lab. I finished my postdoc and started this job here at Sac State. So Dr. Rochelle Tanner is the one who did the assembly and read mapping for this data. So remember, we need to do that assembly and gene mapping, read mapping step to be able to get estimates of gene expression for all of those different individuals across all of these different genes in the data set. And we'll be working with a data set today of about 1,500 genes. And what we'll be doing is creating a PCA with that gene expression data using a package in R that I've used in my previous research called FactoMineR. And just to acknowledge the other pieces that came into getting you this data set, faculty and staff at Hopkins Marine Station, some other labs that hosted us while we did this work, and funding sources, an NSF grant, and including funding to Florian through the SURE program, which is a program for Sac State undergraduates to do summer research. So if you've never heard of that before or might be interested in it, I can tell you more about it if you're interested.